Hello and welcome to your pre-lab lecture on enzymes. We have a number of objectives to get through today, but you will be learning about how enzymes work, what they're made of, what the different parts of the reaction, structure, and then we'll be doing an experiment on looking at the effects of enzyme concentration and substrate concentration. Click here. Next week, you're turning in an introduction section to this lab. There will be a handout and citations to work with. We will have a quiz on this lab, and I'll just keep saying this so that maybe you'll hear it. It would be a good idea to read the introduction to Lab 7 for the following week and write in your hypotheses before we get going. But that's all for next week. Today, we're talking about enzymes. Enzymes are proteins. That is the organic molecule of choice. There are lots of different kinds of them. Anything that ends with that ASE ending is an enzyme. One of my favorites is lactase, which is an enzyme that breaks down lactose, which is the sugar in milk. You may have seen packaging such as this, or on your friendly neighborhood milk jug, such that people who can no longer produce their own lactase can take this and eat dairy. Hooray. Enzymes make reactions happen. They generally do this by speeding them up and by lowering the amount of activation energy needed to make the reaction happen. So in this graph here, we have the purple line is what would normally, the amount of energy would normally take for a reaction to happen. And then the products are lower. With an enzyme, it only takes this much energy. So either being able to do it at a lower temperature or with less ATP input makes it much easier for reactions to happen. Some reactions would be so slow without their enzyme counterpart that it basically doesn't happen. All right, so this is a more moderate graph that we're looking at here. That activation energy is something that you should understand, though, of needing more energy without the enzyme there to help out. Okay. In terms of the structure of an enzyme, remember that proteins have the initial lineup of amino acids, a secondary structure, which then has the sulfide bonds, so that tends to be folding or zigzag pleating. Then we get a tertiary structure of the amino acids interacting with each other, and we often then have a quaternary structure of two or more three-dimensional molecules joining together to form the final molecule. Here is a diagram of a very three-dimensional enzyme, which is represented with all this blue blobby. Okay, and it's important that it has this three-dimensional structure because it has a very specific active site where that substrate fits in to the enzyme molecule. Right? So this is right here is the active site. This substrate is going to fit in here. When the two are together, you can see that the enzyme changes shape slightly. Okay, we call this unit when they're together the enzyme substrate complex. But then the substrate, so something is being done to the substrate here when it is in this position. And then it's released as a product or products, and the enzyme goes back to being just how it was. So it is unchanged by the reaction. The substrate is changed somehow, depending on which reaction we're talking about. All right, here's an image from your book, from your manual. So we have the enzyme and the substrate. That's the beginning molecules that we're talking about, which the substrate fits into the active site of the enzyme. This new unit then is the enzyme substrate complex. In this case, we're joining these two substrates together, all right, releasing water. This is a dehydration synthesis of a new molecule. Those substrates are released from the active site as the product, and the enzyme remains. So in terms of letters, we have enzyme plus substrate here. When they're reacting together, it's the enzyme substrate complex, ES, and the enzyme is released untouched, unchanged, and the products at the end of the reaction. And that is on page 66 in your book if you want to be looking at it some more. An analogy for this is a stapler and staples and paper. Okay, so the stapler is our enzyme, right? We need to load it with staples. There's always more papers that need to be stapled, right? But as you're thinking about this, this is the tool that is making this reaction happen of getting the paper stapled together. The stapler at the end is going to come out the other side ready to staple another paper, right? So once we've loaded this substrate and attached, we can put the stack of papers in and engage the stapler. So that's the enzyme substrate complex there. And then we have the stapler is done and ready to staple some more, and we have a stack of products here of papers with staples in them that are now in packets instead of just singular. Right? So the stapler, though, is ready to go again. It hasn't changed. Just like in an enzyme reaction, the enzyme, although it modified its shape during the reaction and when it was part of the enzyme substrate complex, it is essentially ready to go again. The staple, right, is now different. It's been bent. It's not square anymore, and it's holding together the paper. So the product is a modification of the initial substrates, which are the plain staples and the plain paper. And now the product is a different thing. Hopefully that analogy will help you with thinking this through. So there's our initial stapler is still the same and ready to do it again. There are 2 videos that you might want to watch. I'm not going to play them right now for you, but you can go into the Google slideshow and do it yourself if you're interested. And then just really quickly, I want to talk through the reaction that we're going to be doing in lab. Catalase is an enzyme that breaks down hydrogen peroxide, that's the substrate, into water and oxygen. And most cells make hydrogen peroxide as a waste product. It's highly reactive, though, and toxic to cells, so most cells also have catalase to break it down into innocuous water and oxygen. You may have put hydrogen peroxide on a cut before and notice that it bubbles, so you can see the oxygen coming out. Right? And that's because on your cut, the cells have been opened, right? And the catalase in the cell has been exposed, and then you pour the hydrogen peroxide on it to clean it. It's toxic, right? It's going to kill any bacteria. It's also killing the cells, and the catalase in your cells is trying to break it down into oxygen and water. Still a good thing to use. Right? So our reactant or substrate is the hydrogen peroxide, H2O2. The enzyme that we're talking about is catalase, and the products that are at the end of the reaction are water and oxygen. Okay, so read through the rest of the lab so you have an idea about what we're doing, and we'll just launch right into this basically as soon as we get to class on Wednesday. Have a great week.